Is it working? Isn't the video working? Scroll back and face to you. Excellent. Okay, thanks very much, everybody, for joining us this afternoon in person and online. And welcome to the Centre for Marine Science Postgraduate Seminar. I'm Tracy Reid, and along with Apurva Prabhu and Helena Kurisic, who isn't here today, will be co-hosting today's seminar. And we're very excited to be joined by Jord Wiegering and Dr. Asia Haynes from the University of the Sunshine Coast. Jord is from School of Civil Engineering. Before we begin, I'd just like to acknowledge the traditional custodians of the land from which we join this meeting today. For most of us joining from Yanjin, we're on the land of the Yagara and Turrbal people, and I pay my respects to their elders past and present, as well as the traditional owners of all the regions from which our listeners are joining us today. And some quick housekeeping for those on Zoom. Please keep your video and microphone switched off throughout the presentations, and at the end of the talk, we'll have time for questions. If you have a question for our speakers, please feel free to post it in the chat and we will share it with them at the end of the presentation. Otherwise, we'll call for questions after each talk, and you're welcome to unmute yourself. So first off, I'm very happy to introduce our first speaker, Jord. Jord's research is on the mitigation of sloshing in floating closed containment fish farms. The marine aquaculture industry is expanding operations in floating impermeable tanks at farming sites that are exposed to waves. Tank motions excited by external waves can cause the contained water where fish reside to oscillate, i.e. sloshing. Resonant sloshing causes significant problems. Novel slosh suppression blocks are developed to mitigate sloshing in the design process. Fish wellbeing is considered to create an optimal environment for fish. So please join me in welcoming Jord.
Yeah, thank you. Yeah, thanks. It's great to be talking here today to you. Yeah, so thanks for the introduction. So for those of you who do not know me.
Sorry, can you just share the screen first?
Oh, important. That one. There we go.
Yeah.
Oh,
Oh, you're right. Yeah. Let's see if we can move this one out of the way as well. Is that good? Hide. Yes, awesome. All right, thank you. So for those of you who do not know me, my name is Jord. I am a PhD candidate of the Faculty of Civil Engineering here at the UQ, and my research is on floating closed containment aquaculture systems. What that means and what that all includes, you'll get to know throughout this presentation. I'm an engineer. And I've really treated my research from an engineering point of view. And I'm very well aware that we're putting fish inside here, and there's quite a major overlap in topic with marine sciences and the biological aspects of putting fish in a containment system. And I think this is really where the interesting part is for the marine science point of view. And we'll have a discussion about that at the end of the presentation, because this is where the overlap is. So today my presentation will start off with talking about why are we fish farming in the first place? Why is that a good idea, and why do we need that? Then we'll look at current practices in the marine fish farming space at the moment, and we'll continue with floating closed containment aquaculture systems, and then you'll get to know a bit more about my research, a little bit about the technical aspects, and from there we'll move on to I hope to get a bit of a discussion on how does this relate to the marine science space. So fish farming. So why are we farming fish? And this is a plot from the United Nations, and it gives us an overview of the total amount of fish produced globally. And what is interesting in this plot to see is that roughly from 1990 onwards, that's everything below the red line, blue, that's the wild captured fisheries. They plateau at that point. And that indicates an overfishing and depleting wild fish stock. So to provide the increasing demand for fish, that all comes from aquaculture. And part of that is inland aquaculture and part of that is marine aquaculture. And we'll be focusing on the marine aquaculture side of things. So in Tasmania, it might look familiar to you. This is what aquaculture can look like in the marine space. And on the photo, what you see here are fish pens. And what it really is, it's sort of a hula hoop structure. It's a floating collar, and there's a net suspended on that structure. And what we do, we put fish inside, we feed those fish until they grow to a certain size, we harvest them and we sell them in the supermarket. That's how it works. These systems, they've got a lot of challenges to them. Like the biomass inside these systems is quite substantial. So it's an excellent breeding ground for pathogens and parasites. There's in Norway, this particular to Norway, they've got a big problem with sea lice. Sea lice, they flourish in there and they have quite devastating effect on the fish that we're farming. We've got seals and sharks. They're excellent predators, and they're always in for a fresh feed. And that really affects the stress levels of the fish inside. And I think this is for me personally is something that really strikes me, is that the waste management. So we are using public waters and we feed these fish, and these fish they poo, and that all drops down to the seafloor. And we rely on the local ecosystems to actually take care of all that, all the feces. We've got the water quality is determined by the external water environment. So that's by the oceans itself. So if there have been incidents where there was less oxygen, a body of water traveling through a net pen with too less oxygen, and all our fish, they pretty much died. So there's a novel industry emerging, and that's only from recent years. So that's starting to become something, and that is fish farming in closed containment systems. And this is an impression of that. And what this really is it's a bucket with an impermeable wall, and that's a physical separation between your internal and your external environment. So what we do in these type of systems is we pump in water, we filter that, we ozonate that, we oxygenate that, and we really create like an optimal living environment for fish to grow, because that's the output of our system. We want as much biomass as possible. As if fish die, feces and excess fish feed that drops down to the bottom of the containment, we collect that and we process that, and we don't put that in the ecosystem. There is, however, an issue. There's a limited amount of farming sites that we can deploy these systems in, and there's a few commercial ones around there already. However, there's the issue of sloshing, and sloshing, I would like to demonstrate that over here. So if you've got a containment like this, let this be your fish farm, and you only give that a small movement, you can see that the fluid inside starts to go quite crazy. And this phenomenon is what you call sloshing, and this is typically what you will see in these type of systems when you put them in slightly more exposed sites where you encounter waves. So that's what we're going to be exploring now, that you get an understanding of what are we talking about. So the Norwegians did some experiments. With a model of a closed containment tank system. Then I'll just play the video, then you can see what happens, and I'll talk you through what's going on. So we've got relatively small waves here that excite our structure, but they're excited at exactly the right wave period to excite resonant sloshing. And what happens is that the fluid inside this containment starts to go wild. And that's a problem for our operation, for fish well-being, and the structural integrity of a containment. There's been a case where, like a tank like this burst, all your fish gone, local fishing people very happy, but like your operation, fish farming operation, it's not very beneficial. So in my research, I did similar experiments with a floating closed containment tank in waves. And based on the measurements of the sloshing, you can make a mathematical representation of your internal fluid, and this is what that can look like. So this is the mathematical representation. So if we're talking closed containment systems, we're talking about the scale of these things is about, they have about diameter of 40 meters. And let's say on full scale, we're exciting that at 5 seconds, and this is a sloshing response that we get. Because we mathematically approach what we saw in the experiments, we can break that down into individual sloshing modes and can develop a better understanding of, hey, what is actually happening in terms of these internal fluid oscillations. And I've done that here. And really what you see is that there's only a couple of sloshing modes that are dominant in the overall sloshing response. So there's 3 here. We've got, don't pay too much attention to the numbers, but it's sloshing mode 1, 2, 2, 1, and 1, 1. And there's a little bit of noise that we don't capture and we're not too interested in. But in this particular case, what you see here is that we're exciting at about 5 seconds. And because we're exciting quite close to the natural sloshing period of that sloshing mode 2-1, that's the one that's really dominant in the response that we're getting. So the message here is, if we are farming in sheltered farming sites, we typically have wave periods 1, 2, 3 seconds. And then we're operating in the regime where we're not in the proximity of these natural sloshing periods. But as soon as we move outside of fjords in slightly like bay environments, or slightly like a bit more exposed, we're starting to get wave amplitudes of 4, 5, 6 seconds. And this is bang, that's where you get your sloshing responses. So this is really the challenge that I'm facing in my PhD, and this is like the problem that was posed, and like the question was raised to me, how can we solve this? So I came up with a solution, and that looked like this. It looks fairly simple, but we call it a slosh repression block. It's the green ring. So we're looking at a cross section of a floating closed containment tank system, and this ring has got 3 characteristics. So the top of this ring is flush with the free water surface. So if you get a positive variation of your internal fluid, that will spill on top of the slosh repression block. The depth of the thing is deep enough that you don't get any air trapping or slamming loads, because that's associated with shock waves, and that's not nice for your fish. And then the width of the thing is substantially wide to facilitate some sort of overtopping. So think of it as like a wave on a beach, where the wave breaks, it dissipates its energy, and then it drizzles back. So that's a slosh repression block. And how that works in practice, there's an analogy with a swing. If you put someone on a swing and you give it a push, at exactly the right moment, you'll see that the system itself starts to gain more and more energy. So in oscillatory motion, so if you're sloshing from left to right, like on the left figure, you've got a perfect exchange between potential and kinetic energy. So we gain potential energy, we come down kinetic energy, and then off we go to potential energy again. And we give it a little push, and that increases the total energy in our system. Now with the slosh repression block, you interfere with that cycle. So as you gain a little bit of potential energy, you spill straight onto your slosh repression block. As you go down, the fluid has a horizontal component rather than a vertical component, not feeding that exchange cycle. So this is what I've developed in my PhD, and this is what I've studied in an experimental setup. So my experimental setup looked like this. At the civil engineering facility, we've got several wave flumes, and I made a containment tank model of a fish farm, and this wave flume allowed me to make different types of wave climates and waves and see what the sloshing response was for different slosh repression block width and inserts, and it allowed me to study what the behavior there was of the internal fluid. So part of my test configurations included the following. So we're looking at the slosh repression block where we've got overtopping, and then I made the comparison with what happens now if we do not overtop. So we've got vertical walls there at the free surface where your fluid can run up against you. So this is a video, but first I want to introduce to you what we're looking at. So on the left, you'll see a video with a slosh repression block, and then you'll see that water goes on top of there. And on the right, and that's without overtopping. you'll see that the transparent plexiglass, you'll see the water run up there, and you really see a difference between the sloshing amplitude, and that's also represented in the graph down here. So red is the free surface oscillations with a slosh repression block, and the black one is without the overtopping. So this is sort of what you get. So you really see on that right video that fluid just goes interrupted and starts oscillating. And this is a scale model. So in practice, this will be like significant. I've tested a slosh repression block with a smaller free surface, so a wider width. And what is typical here, that if you do not have overtopping, you see that there's a constant increase of your sloshing amplitude. So this is really where you hit that resonance. And if you allow for overtopping in this case, this is one of the 100 tests or so. But you really see that signal tapers off, and we mitigated some sloshing here. Now that's all great, right? We've mitigated some sloshing, and we did something about the sloshing amplitudes, but there's more to it, because we're not only interested in sloshing amplitudes. We're putting fish in there, and those fish need to be happy. So part of my experiments included like putting dye in the sloshing fluid to actually get an idea of like, hey, what happens in the fluid itself. So before we start the video, you have to take note that if you put dye in the water, it spreads by itself, and that's what it looks like after some time. So that's how, like, you have to take it into account. You have to take that into account when you interpret these results. So the top left is just a containment tank by itself, and that's where we have two large sloshing amplitudes. The top right is a conventional way of damping sloshing, and that's typically what you see in the oil and gas industry. They put baffles in there, and really what a baffle does, it creates, as flow passes a baffle, it creates a lot of flow separation, a lot of vortexes, and a lot of chaos in your internal fluid. And then both the bottom ones are with a slosh repression block, where we have reduced the where we have reduced the sloshing amplitudes. And what you'll see is that the disturbances to your main fluid volume, they remain relatively shallow. And that's exactly what, so that's exactly what we want. So in the top right, you see that there's like the chaos kind of spreads into the complete fluid domain. And then with the bottom two, you see that they remain relatively shallow. So this is in line with how that fluid from the top of the slosh repression block kind of flushes back with that horizontal component, and does not feed that potential kinetic energy cycle. So I think this is the part where there's the overlap with marine sciences. So in summary, we've talked about like, hey, fish farming, why do we do that? We've looked at closed containment systems. We've looked at the sloshing problem, and we've looked at a possible solution to overcome this. But now we've got fish in there, and those fish, they need to be happy. Like, a fish farmer is interested in one thing, and that's like, how much feed I put in and how much biomass do I get out. So as we move these tank systems to more exposed waters, there is tank motions. These tank motions, they trigger sloshing. This sloshing causes disturbances. But I've not been able to find any quantitative data on like how happy, or like from a biological point of view, this is how I would say it, like, how happy are my fish in my containment? Like in doing research towards these type of systems, you make the assumption that there is a certain tolerance to certain tank motions. It's a bit like motion sickness in a car. Like, when do you get motion sick? But yeah, so I think this is, because this is a novel industry, I think there's a lot of potential there for future research to actually address this. So where I've been working on the engineering side of things, there's also the biological side of things, and that's something that's unaddressed at this point. So that's where there's a lot of potential. And with that, I would like to finish off. Yeah, yeah. Yeah, so if you've got questions, comments, or anything really, then yeah, I'm more than happy to answer. And yeah, thanks for the attention. Yeah, of course.
I know you just said like you don't have any quantitative data to say like if the fish are happy or anything, but did you have any requirements of you need to be able to let X amount of sunlight in, or like there needs to be like X amount of surface area or anything like that to be able to keep the environment quiet or anything like that?
It is like fish farming, maybe the online. So the question is like, what is the minimum required free surface area?
Yeah, or just
Right,
did
what-
you
what's-
have any
what-
type of-
No, it's very much a practical interpretation. Like there's a fair few commercial systems out there, and some operate like I actually did the math on like, okay, what's the volume of my containment? And fish farms generally run with a fish density of 25 kgs per cube. So that you've got 1 square meter, you put 5 full grown salmon in there. That's the equivalent of that. And then I took the ratio of like what the free surface area was per biomass kind of thing, and you see that you can go quite small. And salmon in particular, they need free surface to fill the swim bladder. But yeah, like there is room there to contract that free surface with slosh pressure block. That's what I know. But how much that is, that's something that's also not quantified. Is that along the lines that you? Yeah. Yeah, but I would love to know that. Like in Norway, they're farming in net pens, and they've really got that problem with sea lice. So what they tend to do is, sea lice, they sit in the first 10 meter of water column. If you go lower than that, they apparently don't live there or sit there. I'm not sure how that works, but they designed sort of a tube cage, they call it. So they've got a net system, which is sitting lower. And then they've got a sort of a snorkel tube with a sort of a wall. So it's still a net cage, but they barrier off like the first 10 meters. And that is also a contracted free surface. They're also doing experiments with what happens now if you completely submerge this cage and have a sort of a dive bell in there. Like it's a bit like a upside down plate or upside down bowl where they pump in air, where the fish should be able to kind of grasp air there. But that is relatively small to your contain, but that's all like in a juvenile stage of development, really. Yeah. Yeah. But, okay. Chat. Let's see. Oh yeah, that's the bottom one. So in the video with the dye you showed, it seems that no slosh damping worked better than the traditional approach. No slosh damping.
Sorry, I asked the question. Can you hear me?
Yeah.
Yeah. Just the video you showed with the 4 squares, and it had the there was no slosh dampening in the first one, and then the next one was the traditional approach before you showed your 2 examples.
Yeah.
It looked like the first one didn't have as much sloshing as the traditional approach. Is that right, or did I misinterpret that?
Yeah, that you misinterpreted that.
Okay.
Yeah. No, if you do the measurements, then you'll find that there's like less sloshing in the conventional damping one.
Oh, okay. Okay.
Yeah.
It just seemed to dissipate really fast in that one.
In the one without the damping.
I thought in the conventional one, but anyway, sorry. I might have misinterpreted.
Let's go to the video. Oh, I zoomed in. What do I do now? Yeah, so you're saying
on the top line, that first square.
Yeah.
Oh, that has conventional dampening.
Yeah.
But the top right doesn't, or?
No, so the top left has got nothing. There's got nothing in there, but just very, very, very big sloshing amplitudes. And the top right has got conventional damping, so reduced sloshing amplitudes, but a lot of chaos as a result of that.
Yeah, right.
Yeah.
Okay.
Thanks for your question. Thanks for your question. Yeah. Yeah.
So you put in these shells around this.
Yeah.
And obviously once they're scaled up, they're big. Is there a risk of fish being so densely packed they're probably going to jump out and stranding themselves in there?
Yeah, I like the question. So the question is, don't the fish jump onto this slosh repression block thing? And I've been active, like from this is more of a personal thing. I've been active in fishing and handling fish in aquaria and whatnot. I've never seen a fish jump on a beach, and they know that it's there. Like that's my first. That would be my first answer. But like a second answer would be, you can net it off, like where just water flushes through. So I don't see any issue there. Well, yeah. So like in conventional fish farms, you've got like the net cages, and you've got a tube there, and there's also a net on the outside to prevent them from jumping out. So I can imagine if this gets developed in one way or the other, then that would be a way to mitigate that. Yeah, thanks.
Is there any data on the natural levels of turbulence that the aquaculture species experience outside of the name?
It is.
In dealing with sort of semi-aquatic fish, at least at the time that they're being farmed, and they're likely to experience less turbulence, less chaos than, for example, reef fish, where waves are constantly crashing.
Yeah.
I'm just wondering if there is any data on the natural levels of sloshing / turbulence that fish species experience.
If so, I'm not aware of it. Yeah. There is a fair bit of research on, like fish farming in net pens is really big. So they've looked at what is the critical swimming speed of a fish, or what's the maximum current that we can put these fish in. And as you put flow through a net, you'll get like some sort of turbulence shredding from those nets happening. Yeah, but no, it's not quantified, and I guess the sloshing is this is more related to sloshing. Sloshing is a planar motion of fluid. So if you look at the particle trajectories, they're all linear, and in the ocean it's all orbital and influenced by waves. And I've got no clue what would happen to a fish if you put that in sort of a piston type of. So it's like piston type of motion.
Qualitatively different type of the experience of sloshing.
Yeah. Yeah, it's like orbital versus linear. And that's, yeah.
It seems like the most natural metric as to how much you have either rate of breeding or rate of mortality. Do you ever get that information when evaluating your methods? I mean, you're working in obviously a normal habitat fish. I imagine you're actually evaluating it. Maybe
yeah, it's nice that you bring that up, because I've had a chat to Andy Barnes. So I don't know if he's familiar to you, but he's like the marine fish expert here. To set up an experiment with closed containment tanks in the wave flume in the civil engineering facility, put like juvenile salmon in there, feed them, and like do the experiment and have a control group next to that. And I find myself like not being a biologist. Yes, I find it very interesting, but we made the assumption, okay, listen, okay, I'm an engineer, so we're just going to focus on the engineering side of things. Yeah, but there was like cortisol levels and all sorts of things involved there. Yeah, thanks.
So thank you again, George, for presenting.
Yeah.
Okay, so I'm very happy to introduce our next speaker, Dr. Asia Haynes. Asia's research is focused on the biodiversity of Lady Elliot Island in her project known as Leaf to Reef. Lady Elliot Island is located at the southern end of the Great Barrier Reef and has been identified as a wildlife refuge from the impact of climate change for migrating tropical species. One of the aims of the Leaf to Reef is to assess the biodiversity found in and around the waters of Lady Elliot Island and examine how connected these waters are to other parts of the Great Barrier Reef and beyond. So please join me in welcoming Asia. She's presenting online, so just bear with me a sec.
Would you like me to share my screen, Tracy?
Yes, do I need to enable that?
Replace pin, it should pin it.
Oh, yeah. Sorry?
Yeah, that one.
Yep. Okay, that's good. Okay, ready to go.
Okay. Oh, well, hi, everybody. Sorry, I can't be there in person. And thank you for the intro, Tracy. Is the sound okay there? Can everybody hear me all right?
Yes.
Yep, fabulous. Okay, so I'll just expand on the earlier acknowledgement of country by acknowledging the Midjinbul people of the Bundjalung Nation, the traditional custodians of the land on which I reside. And I'd like to pay my respect to their elders past, present and emerging. And I'll also extend that respect to all First Nations people, including those attending today, and acknowledge their continuing connection to land and sea country. All right, so the Leaf to Reef team is a very big one, and it includes researchers from the University of the Sunshine Coast, University of Queensland, Southern Cross University and Flinders University. The project is led by Dr. Christine Dudgeon and Associate Professor Kathy Townsend is the academic lead. And then there's the whole collection of people involved in the various aspects of the research, including a number of PhD and honours students. And I'm involved with the project to manage the biodiversity data, assist with data collection in the field and conduct outreach and engagement activities. All right, so I guess I'll start with why Lady Elliot. So Lady Elliot Island, as Tracy said in the intro, is at the southern end of the Great Barrier Reef, and it was the first climate change arc that was included in the Great Barrier Reef Foundation's Reef Island Initiative program back in 2018. And it's been identified as an important land and seascape in the face of climate change for four main reasons. So firstly, its conservation value, where it's likely to act as a collector of climate refugees from the central and northern Great Barrier Reef. Second, for its biodiversity, it's in a unique location close to the edge of the continental shelf upwellings, which attracts not only reef but also oceanic species. Thirdly, it's of economic importance, home to the multi-award-winning Lady Elliot Island Eco Resort. And lastly, due to the threats it faces in terms of invasive plant species, coral bleaching and crown of thorns. So there's three main aims of the Leaf to Reef research. The first is to generate high resolution maps of the island and the reef flat. The second is to understand the impacts of the extensive revegetation project on the island. And the third is to quantify the biodiversity of vertebrate life on and around the island and determine its connectivity to other locations. So for the purpose of today's talk, I'm just going to be focusing on aim 3. But I'm more than happy to answer any questions people may have about other aspects of our work. So the reason that Lady Elliot Island is of particular interest in relation to climate change, I'll just start by taking a look at some output from a paper that some of our team were involved in a few years back. And here we can see that global predictions of species range shifts, with the blue areas indicating species loss and the red areas indicating species gain under two different climate change scenarios. So business as usual on the bottom and aggressive mitigation on the top panel. And as you might expect in both scenarios, the tropical areas are predicted to lose species, while the subtropics and temperate zones will be gaining species. So our team ran this analysis in high resolution for the Great Barrier Reef to reveal that the area around Lady Elliot Island, located in the black circle, is expected to either gain species or maintain its species diversity under future climate scenarios. So this means that the waters in this region may represent climate refugia for thermally driven range shifts of mobile species in response to climate change. So the first part of this aim is focused on the biodiversity found around the island. So to monitor change into the future, it's vital that we understand what's there already. But despite Lady Elliot being a popular tourist destination and part of the well-researched Great Barrier Reef Marine Park, there's not currently a species list for the island and surrounding waters. So what our team set out to do back in 2020, when this project started, and we're doing this using a variety of approaches, so including dedicated research surveys, assessing online databases, and encouraging citizen science submissions. So this is just an update on where we're at with this aim. So bearing in mind that we're only focusing on the vertebrate species here, no offense to coral people or invert people in the audience, we just didn't have the capacity or expertise to tackle the invertebrates. So we've now catalogued close to 600 species around the island, with the vast majority of these being the teleos or bony fish. And one of the cool outcomes of compiling this biodiversity list for the island is that we intend to create a field guide for the island and surrounding waters with the help of some legendary fish nerds and taxonomists, Mark Erdman and Jerry Allen. But also bringing Mark onto this aspect of our research was a little bit serendipitous, as last year Mark joined us to conduct some manta ray tagging off the island, and it opened up some discoveries we kind of hadn't planned for. So in between tagging manta rays, Mark started looking at the other fish on the reef, and he pointed out up to 6 species that he believed might be new to science. So 2 of these had been staring us in plain sight, a couple of rather common damselfish species that have potentially been misidentified in the past, and which he thought warranted further investigation. So our most recent fieldwork in February, we collected a number of specimens to conduct morphometric and genetic analyses of the 6 species of interest. And yeah, you can watch this space for the outcomes of those investigations, but it looks like we might have a number of new species to describe. So that's pretty exciting. Okay, so moving on to part 2 of this aim, which is connectivity. So in order to assess the resilience of Lady Elliot as a potential refugia for migrating species, we also needed to understand how well connected it is to other places in the Great Barrier Reef and beyond. So for this part of the project, we've selected several key species of interest to conduct tagging and genetic studies to determine the movements and connectivities of these species to other reef systems and to habitats further south. So today I'll just go through some examples of the outputs we've collected so far. So the first example we'll look at comes from the island's red-tailed tropic birds. Lady Elliot Island is fortunate to be one of only 2 locations on the Great Barrier Reef where these elusive birds nest, and the other is Rain Island in the far north. And our population of interest on Lady Elliot is the youngest and smallest known red-tailed tropic bird population, having colonised the island less than 40 years ago and consisting of only 6 breeding pairs. So this study was led by an honours student, Caitlin Ivey, and compares the genetics between Lady Elliot Island and Ashmore Reef to provide insights into gene flow and connectivity between seabird populations separated by thousands of kilometres. So this is some of the results from that study, which identified a weak signal of population structure across these locations, but also evidence of gene flow. And the conclusion was that these breeding colonies are likely part of a much larger population. And the study found really low levels of relatedness and inbreeding within these subpopulations. And this is likely due to high gene flow preventing an inbreeding depression. So these tropic birds are also of interest as they spend most of their life at sea, but during nesting time, they take turns feeding their chicks, and they represent an important source of nutrients for the island. but we've actually got no idea where they were going during their foraging excursions. So a couple of years back, we deployed some satellite tags on the birds and managed to capture a fantastic 36-hour track from one of the birds that demonstrated foraging close to the shelf edge in proximity to the mesoscale oceanographic feature of the Capricorn eddy in this region that's a source of productivity. Okay, so the next example are the Capricorn silver eyes. So these cute little birds first colonised Lady Elliot Island in the 1970s after the vegetation returned to the island. And we had an honours student, Annika Radu, examine genetic population structure between Lady Elliot, Heron Island and the mainland. And she found that despite being very close to Heron Island, that the birds from Lady Elliot are genetically distinct. Although you can see here, there's a little outlier between those populations, which suggests there may be some genetic exchange starting to occur. However, the mainland population was clearly separated from these island locations, which suggests minimal connectivity between reef-based populations and the mainland. Okay, so on to the elasmoranx. So the next example comes with the epaulet shark, which is one of the top predators on the reef flat and is a species endemic to the Great Barrier Reef. And this work is the result of an honours project conducted by Jacinta Shackleton. And the study investigated the variation in body patterns and genetic structuring of epaulets throughout their range, with data sourced from 20 locations along the latitudinal gradient of the Great Barrier Reef. So here's some of the output of these analyses, the body pattern analysis on the left and the genetic analysis on the right. So in brief, the body pattern analysis found significant differences between most regions, particularly between the far north in the dark blue here and the south in the red. And then there was some overlap in the middle with the central and northern Great Barrier Reef sections. And then on the right, the genetic analysis found 4 distinct populations as well, which complemented our findings from the pattern analysis. So this is a really exciting combination of techniques used to explore their connectivity. And then the next example are the leopard sharks, and this work is led by Dr. Christine Dudgeon. And for this animal, we conducted field work off North Stradbroke Island, as this is where they aggregate in large numbers during summertime, so it's easier to access the animals. And we collected animals on free dive with the very talented help of freediving extraordinaire Cam Cotterill, for those of you who may know him from MBRS. And the sharks were brought onto the boat and had water pumped over their gills while measurements, genetic samples and bloods were collected. Then the shark was internally fitted with an acoustic tag with a 10-year battery life for tracking. So for those that don't know, acoustic transmitters get picked up on acoustic receivers located across the reef, and the tagged animals need to be within range of the receiver, which is about 500 meters to a kilometre. And then the receiver needs to be retrieved, the data needs to be downloaded and then uploaded to the IMOS database to receive information from across the network. So here you can see the extent of the network across Australia, but particularly in Queensland. And one of the important considerations with this data is that it is presence only. So we don't have information about where the animal is when it's not within range of a receiver. So back to the leopard sharks, what we saw from the early movement data shows them moving around the southeast last summer into spring, with the FAD sites corresponding to fish aggregation devices located around the Gold Coast. But unfortunately, the data from last year will need to be downloaded from the receivers and uploaded to IMOS portal for us to update their known movements over the past year. So we haven't recorded them up on Lady Elliot as yet, but we're getting there. Okay, so then the next example is of detections of a tiger shark named Amy that was tagged internally at Lady Elliot in 2020. And the top 4 lines corresponds to detections at sites around Lady Elliot Island, showing really good residency to the island with just a few detections down to the Gold Coast on those FAD receivers. And then the final shark we tracked at Lady Elliot is the grey reef shark. And here we can see some incredible residency to Lady Elliot. So given this is acoustic data, we don't know where the shark is between detection times, but there's quite decent stretches where it appears the shark doesn't leave the vicinity of the acoustic receivers around the island, and it hasn't been picked up anywhere else as of yet. Okay, so the last species I'll talk about is one of the closest ones to my heart, the manta rays. So we've been studying their connectivity with Project Manta around Lady Elliot Island, well beyond the life of the Leaf to Reef project. But we've also gained some interesting recent insights into their movements here as well. So one of the reasons for this is that we've been fortunate to deploy some of these fancy satellite tags for high resolution tracking data. Let me see if the video plays. Yep. So here you can see the deployment method is by hand spear with the tag attached to the dorsal side of the animal. And it's programmed for an average 100 days attachment. So unlike with the sharks, which we can catch and implant internal tags on, manta rays are too big and awkward to do this with. So we rely on external tagging measures, which do not have nearly the same retention times as the early internal tags. So here's the output from 5 of these tags, all deployed at Lady Elliot Island over the last couple of years. And I just wanted to play this video as well to show that the animal with the tag attached, even though the tagging process does look like it really disturbs the animal, they often return to their same behavior within a couple of minutes. So this is that animal returning. You can see the tag floating above it there in the video. So we can see from the tracking data that the majority of the rays spend quite a bit of time in vicinity to the island, but some do move up into the other Capricorn Bunker Group islands, and some spend quite a bit of time off the northern tip of Gari or Fraser Island in the Long Shoals region. And one of them spent weeks around the beaches of the northern stretch of the island. And this is quite an exciting finding for us, as we didn't know that they spent time in this region. And the tagged ray with the orange track was heavily pregnant. And so we wonder if this points towards potential pupping habitat, which is unknown for this species. Okay, so moving on now to some acoustic tracking data for manta rays. We tagged 10 mantas with acoustic transmitters, and in the following months, the tagged animals were picked up throughout the Capricorn Bunker Group at Heron Island, Northwest and One Tree, which complements our data from the satellite tracking. And then another way that we track manta rays is via photo identification, using their unique spot patterns to track individuals over time and space via photographs. And the awesome thing about this technique is that citizen scientists can get involved, which can really extend the spatial extent of records for understanding range, movements, and connectivity. So using this method, here's just an example of an exciting finding from earlier this year. So the green lines represent movements made by some mantas a few years ago between southern aggregation sites, including Lady Elliot Island, all the way up to the SS Yongala, just south of Townsville. And then this year we received a sighting of an animal from near Cairns that had previously been seen at the SS Yongala. And this is the first record of connectivity between sites farther north and south, which we had previously assumed may be separate populations. All right, so I hope I've given you a good insight into the different work we've undertaken to address these questions around biodiversity and connectivity. And with that, I just wanted to thank all of our funding partners and once again acknowledge the large team involved in this work, and then just open it up to the audience for any questions.
Who's got a question? What's that? Okay, so we can't use it at all? No, just have to speak loudly. Any questions in here? Oh, was that? Can she see that?
Yeah, just let Asia know there's a question.
Okay, Asia, there's a question on the chat here. Do
Oh, yeah.
Leaf to, yeah, okay.
Do Leaf to Reef have an iNaturalist page that citizen scientists can contribute to? Bella, that is an awesome question, and I think that would have been a really good thing for us to do in the early stages of this. We're actually, as of the end of this year, we're kind of wrapping up that side of our work, although we may continue depending on funding. I scour iNaturalist to make sure that any species that get posted on there we have in our database of species, but no, I wish I had have started up an iNaturalist page. Thank you for that question.
Any other questions? Yep.
Thanks for your presentation. In one of your slides, you showed quite a small fish that you regarded as potentially a new species. How did you catch that?
So the fish were caught generally using like a aqueous solution on scuba dive and then with nets. So it was all done under ethics and permits and all of that kind of thing. Lady Elliot is a green zone, so we were treated quite strangely entering the boats on that island with fishing gear. But yeah, aqueous solution and small nets.
Any other questions online? No? Okay. So I just want to thank George and Asia again for presenting their research today, and also to our in-person and online attendees. Our next seminar is scheduled for Wednesday, May the 10th at 1 p.m. again in here. And if you would like to present your research at one of our upcoming seminars, postgraduate seminars, please do get in touch. We are looking for more people to speak. So thank you again, everyone, and we'll see you next month. Great. Good. And
Yes.